everyone, and welcome to a new episode of our series of technical interviews. I am Alvaro Guzman, and I am part of the editorial staff at nutriNews International. On behalf of our channel, I want to welcome today's guest, Dr. Todd Callaway, who is an associate professor of ruminant nutrition and microbiology at the University of Georgia. Dr. Callaway, thank you for being here with us. It is a pleasure to have you here.
It's my pleasure. Thank you.
Okay. Before we start, could you just tell us a little bit about yourself, about your academic background, and about your current role at the university?
Okay, I grew up on a real small farm of beef cattle and horses and some leftover dairy cows in the middle of Georgia, and came here as a student to the University of Georgia, did my bachelor's degree. Like most people that get into this, I was planning to be a veterinarian, and I discovered being a vet was the same thing every day, but nutrition was really a key thing of how we could produce animals, how we did things really well, and how we could improve feeding animals and effects on animal health, human health, food safety. So I drifted into ruminant nutrition and worked with a professor here at Georgia for my master's degree. I thought that was really neat. I took the opportunity as an undergrad to do research in a lab, learned, okay, I'm kind of good at this, then did my master's, and was thinking about going to work for the industry for my PhD, and I got the industry instead of going on. And I got an offer to go to Cornell for my PhD with the guy that was the leading rumen microbiologist in the world. So I decided I was going to take that, went to Cornell for 3 years. It was cold, miserable, lots of snow, but I learned an incredible amount. And when I was done with that, I moved to College Station, Texas for the United States Department of Agriculture, the Ag Research Service, and had worked with what's called the Food and Safety Research Unit. Our mission was to improve food safety in cattle, swine, and another one of our groups looked at poultry. So we worked across species on ways to improve food safety on the farm. I did that for about 17 years and then became National Program Leader of Food Safety. And that really, I missed being at the lab bench a great deal. ARS was a fantastic agency, great place to work, but I really missed being at the lab bench. And I got the opportunity to return to my alma mater after about a year as national program leader. So I took it and came back and have been here at UGA for about 6 years now. And most of my research is focused on understanding the interface between the animal and the microbes, primarily in ruminants, but we also work with pigs some and with poultry as well. So how does this microbial population work? How does it function among all the members of the microbial population? How do their end products impact the host animal? How does that affect food safety, availability of food, and sustainability of animal production? So it kind of is this holistic area where I sit in the middle of a lot of things, and I always tell a joke that I tell the nutritionist I'm a microbiologist. I tell the microbiologist that I'm a nutritionist, and you don't let them talk to each other. And that's kind of been my whole career, is living in that space in between those two areas.
Okay. Quite a interesting path. Very, very nice and very enriching previous experiences. I think you're the right person to talk about the topic today. So thank you for your time.
Awesome.
So I wanted to ask you first, what role does the gut microbiota play in the quality and safety of animal-derived foods?
Well, it plays a very large role in that. How large, we still don't truly grasp, because a lot of what we're doing is we're trying to understand how an ecosystem of these microbes exists within another ecosystem, the animal. And trying to separate those two impacts has been very difficult. There's some work we're doing with a company here in Georgia called Kelly Products, where we have a partnership with. What we're trying to do is sort out how much of the performance that we see in quality of the animals, like marbling in beef cattle, how much of that is genetic versus how much of it is that environmental impact from nutrition. And what part of that is handled by the microbial ecosystem. So we don't have a number. I can't say 40% or 60% or 90% at this point, but that's what we're trying to get, is to understand what is the scale and scope of that impact. But it really looks like you can basically make or break the animals with that microbial population. If you don't have a good one, your best performing animals aren't going to be worth anything. But you can make really crappy, genetically speaking, animals with the right microbial population, make them look like good animals by feeding them the right way and having a good, proper microbial population. So it's not the animal dies necessarily without it, but you sure can screw up some good animals by not having proper nutrition or microbial coverage.
Okay, yeah, that sounds good. And in regards to the nutritional side of things and how we can modulate that gut microbiota, what are some of the current products or innovative or new feed additives being used or available nowadays that are aimed at enhancing gut microbiota functionality in food animals? What are some examples?
Well, right now it's kind of a golden age in what we call alternatives to antimicrobials, because antibiotics are becoming less viable, and in some cases they're banned from use as growth promotants, or very highly restricted in their use. So we've started trying to find other approaches, but there's a lot of phytochemicals, of feeding active phytochemicals. The umbrella of what we call direct fed microbials, which includes now the term eubiotics, which are what we think of as these traditional probiotics of a living organism, postbiotics that are the end products of fermentation, like what we would call an old-fashioned yeast product or fungal product. And you also have prebiotics, and the lines between these groupings can be kind of blurred. Like one of the yeast products is a postbiotic, but it may contain prebiotic type compounds. So you'll see a joint effect, and that's called symbiotics, where you've got synergistic, or you add a live organism and a prebiotic, where you give that live organism an advantage. So there's a lot of different approaches that are being juggled with to try to alter the microbial population. There's a lot of companies involved in this. Several companies that I know of that are doing that whole spectrum products.
Okay. And from what you've seen so far, and in the trials that you have carried out, or from other research that you may be aware of, how can these different products impact overall microbial composition and the diversity or the composition within an animal? And how can this impact that animal's overall performance, for example?
Well, that's the multi-million-dollar question that you're asking there. Excuse me. Well, it varies from product to product based on what they are made of, how they interact, the animal that you're trying to work on, whether you're looking at a beef cow or dairy cow or dairy on beef interaction, pig, whether you're talking about a lactating cow or a growing steer, whatever your production phase, because those animals are going to have different nutrient demands, and by different breeds, different environments. So we've got friends that work with a company, and their product, I believe in their product, it works great 99% of the time that I've seen. But if you take it from where it's working here in the Southeast and throw it out in the desert Southwest, their product does not work at all. Why that is, the animals are roughly the same genetically, but it's an environmental effect. So why is there that difference? And as a microbiologist, that's kind of where I'm like, why are those different? Why do they behave so differently in different environments? The environment of the gut is the same inside these animals, but there's something very different. So you have to look at regional, there's a whole bunch of other factors that underlie whether a specific product works. And unfortunately, we don't know what those are at this point. We're still trying to understand how all those work together, how those different factors impact one another, and how they can be layered on top of each other. So it's frustrating in some regards. It's exciting in others, where you can look at it and see what happens if we do this. So I wish there was a way to say this is the best product. I've got a handful of products that I've looked at that I'm like, these are great things, or some that I'm like, yeah, no, don't use that. But for the most part, they all have a role to play, a lot of these different approaches. I'm a big believer in direct fed microbials, everything from eubiotics, probiotics, and prebiotics and postbiotics. We also done a lot of work with organic acids, and that's a really neat way to modify that population. And there's a company that I know that I've worked with a lot from Italy, where they use natural botanicals, or so herbs, phytochemicals attached to those organic acids, and they work really well in modifying the gut fermentation excuse me, and impact animal performance and food safety. So there's a lot of things going on in the space, and it's kind of hard to keep up with, but kind of the rule of thumb I have is the more you look at a company's R&D budget versus their advertising. If their advertising budget is more than their R&D budget, odds are pretty good they're not going to be a great product. And so you kind of got to look at that. But there are some fantastic products out there around the world.
Okay, yeah, that's pretty encouraging. It's, in a way, it's a nice field to be working in and motivating.
It is. And the old days were, where we looked at, there were so many products that were basically magic foo-foo dust that people would tell you, you feed this to your cows, they're going to have shinier hair coat, their teeth are going to be white, they'll learn to drive your tractor, they'll run the feed wagon for you. All those things, those companies, a lot of them have now disappeared. There's still several fly-by-night corporations, but they're not as common as they used to be. So it's really nice to be in the space where the science actually matters to most of these people, and the product working is important.
Yeah, that's sounds good. And in regards to, well, the possibility that these products have to not only impact overall animal performance, but how can they also work to improve the quality and the microbiological safety of foods of animal origin? How can these different products have an impact on that?
Well, that's kind of right in the heart of what most of my research is now. So how it can impact the end product, and the easiest example is looking at beef cattle. If we can increase the amount of feedstuff that is being converted instead of to lactate, but to propionate in the ruminal and the hindgut fermentation, propionate's more energetically efficient for the animal to utilize. They get more ATP per propionate than they do from acetate. Excuse me. But propionate is also involved in marbling. So it's utilized by that tissue for marbling. So if you jack up the propionate level, you're going to get a lot more marbling in your carcass. You're going to have higher carcass quality. And that's a really neat thing. It's one of the things that we're trying to work on a lot with our research, and how it can affect the food safety or human health impacts. The gut is this big ecosystem, and there's niches in it for all kinds of organisms. Salmonella, Campylobacter, E. coli, O1577, and the rest of the interhemorrhagic E. coli's, they live various times in all of our food animal species. And in a lot of cases, they act like a commensal organism, that they're just there. They don't really harm the animal. Some salmonellas, campys may, but in general, they don't really hurt the animal. So they're just passing through, or they may just be this normal member of the population. So if we can find a way to stimulate their competitors or inhibit their ability to compete in the gut, then we're going to drop their populations in that ecosystem, which means you're going to have less carriage into the slaughter plant, which means less exposure to cuts inside that plant. So that's the whole idea, is we want to reduce their competitive fitness and increase the competitive fitness of organisms that make more propionate or more acetate end products that the animal can utilize. So we're trying to kill two birds with one stone. Excuse me.
No, that's fine. And in regards to the research you have conducted, have you looked into or evaluated the effectiveness of some of the products used to modulate the gut microbiota in a long term? Have you been able to assess those impacts like more in a long-term basis, or are you still like in the starting phase of doing different trials with this type of products? And what have you seen, or what influence has it shown on microbial composition in the long term and the quality of the products derived from those animals?
Well, that's a real sore point right there. I have done nothing that's long term. I don't know anybody that really has, simply because when we look for, say, grant funding, people, companies, and government agencies don't want to fund a 4-year study looking at the long-term effects. That's the academic interest, because nobody's going to feed animals this stuff that long very often. I mean, you'll have some cattle producers, say here in the Southeast, that are cow-calf producers that would. But there is no interest in anybody other than nerds like me who want to understand that. So no one has done that to my knowledge. We need to understand that. You're absolutely right. That is something that we need to look at and understand how long it works. But the really scary part is, we've used antibiotics now for 75 plus years. We really don't know why they improve the growth rate of animals. Why feeding tetracycline at 10% of a therapeutic dose, why does it improve growth of chickens or pigs? We have no earthly idea. So a lot of what we need to do is understand how those worked so that we can replicate those impacts. And then that, because we know the effects long-term of antibiotic treatments, can we then extrapolate backwards from that? So I think that's where we need to go, but trying to get that done, get it funded. And, it's like everything else. There's no money, there's no science. It's hard to do that.
Yeah, I totally understand that. So that kind of leads into another question that I had regarding what are some of the potential challenges associated with trying these new treatments or products and evaluating their impact or their influence on microbiota functionality. Is it, from what you mentioned, a great deal of it is from the funding side, but are there also regulatory frameworks that may step in the middle, or is that still like something that hasn't been developed? What's your take on this?
Well, most of the things that are either in the industry or close to the industry, the industry is pretty slick in how they deal with things. They're not going to propose, or at least not very often propose, let's add this product to the feedstuffs, and then let's have to take it all the way through the process of FDA approval. Most things are either on the generally recognized as safe or GRAS list, or have a long history of usage and are approved. Years ago, one of my colleagues came up with a great idea of using sodium chlorate to reduce foodborne pathogens. And I got to see that whole process of it going through the USDA. We're in now year 20, or in through the FDA. We're in 2023. We started that in 2000, and it's still not approved for use. Even though the FDA says, well, there's no downsides to it, but you've got to have a teamwork with companies and a lot of push to get something into the market. And that's a real challenge. So bringing something novel is going to have a lot of pushback and take a long time. So most things are not going to be hampered at that regulatory end. They're just, okay, it's a grass product, let's go. So then you run into the effect of, are you choosing the right dose? Is your diet your animals are on sufficient to get what you need? Are you going to be able to see a response? What's your response that you expect to see? What are you going to want to see out of the animals? What metrics do you want to look at? And designing an experiment like that, it's like trying to throw a punch. And then you start saying, well, I can add this, this, and this. And before long, you've got your force to spread out to be able to make a real difference on understanding the scientific impact of your product. So it comes down to logistics as much as anything when we're trying to make a design a real study that's going to make a huge impact. But yeah, it's going to depend on what your product is, what your animals are, what your goals are. And that becomes really challenging very quickly.
Yeah, I understand. And do you believe another challenge can be that sometimes some of the research that's being done is maybe needs further divulgation, or for producers to learn about it and make them like more willing to try some of these new products, or maybe get them to try some alternatives that at first might be a little bit have higher costs, but these might change as more people start to adopt them. So do you believe there's a need for research to be like trickled down to producers, or do you think that information is already out there and it's more a thing of people taking the leap and going for those products?
Well, it's a little bit of both. Here in Georgia, we have a really good extension service that takes scientific information and boils it into a form that a producer can understand. And one of the things a lot of scientists run into, they're really frustrated with, oh, the producers don't care what I do. Well, part of it is they don't understand what you do, but part of it also is they're busy. They're very busy people. Nutritionists are very busy people. And you've only got so much bandwidth to deal with everything that you encounter in the course of a day. And hearing this great theory of, well, this product will work. Well, they've heard that a dozen times. But until they see the evidence that it works, until they can say, all right, this person that I know or this person I believe used it and they saw this improvement, or you can tell them, here's going to be your return on investment. You spend $10 on this product, you will get $30 back. If you can show them hard data that makes sense to them, my going to a farmer and saying, okay, this is going to increase propionate in your cows, that means nothing to him. But being able to say, we're going to improve your carcass quality by 5% or 10%, that makes a difference in his back pocket. And especially here in the Southeast, most of our producers are legacy producers. They work a regular job. They inherited their land. They inherited their herd of cattle, and they work those cows in the evenings and weekends. So they don't want to spend a lot of time. So that energetic investment is low. So you've got to boil it down to the most salient point for these folks, because again, they're busy. I mean, they don't have time to sit down and read a scientific paper and understand that.
That's true. And in this sense, and kind of adding to this, is how can these treatments, or how do these alternatives compare to traditional methods like antibiotic growth promoters that we all know we need to reduce or completely ban for food safety reasons, for public health reasons? So how do these treatments work in terms of effectiveness regarding achieving the same performance levels, as well as the quality and food safety in the products derived from those animals?
Okay, that's a great question. Again, that's something like part of the World Organization of Animal Health. I help with them and Food and Agriculture Organization. So we do a lot of interest in that. And I worked a lot initially with ionophores, years ago as a growth promotant. And those things are fantastic. They don't really cause antimicrobial resistance. They've been unfairly lumped with true antimicrobials that are used very often in human medicine. So we're working to try to get those back in our arsenal. Excuse me. But the truth is, the products like probiotics and these other approaches, why they weren't used 70 years ago is just consistency. If I make a chemical compound, say tetracycline or ampicillin or something like that, I know what its composition is every time. Consistency is the same. I can then also vary the dose because I know what the active ingredient is. That's harder with probiotics or organic acids because we don't necessarily know always how they work and why. So that makes it a little more difficult to compare apples to apples. But the big issue is their consistency, is we don't know why these work sometimes and why, say, in the desert Southwest, the same product doesn't work. We don't understand why there are those failures, and you don't see that with antibiotics. So that is a real challenge, and that's something that just kind of makes you want to pull your hair out of frustration at trying to understand how can we replace antibiotics this way. And we're slowly getting there. We're getting more appropriate understanding of how these work and actually how antibiotics work, so we can compare and contrast. But it's a slow process because it's really subtle differences. So these products, for the most part, some of them do outperform antibiotics under the right conditions. But then you put it in the wrong condition and it does nothing. And I can't tell you why it's done that or when it's not going to perform right, which is, for me, is extremely frustrating.
Yeah, it's quite mind-boggling because maybe in the trials they seem very promising, but then you don't get them to work always. So it's also difficult for producers to adopt them or in different conditions. So
yeah. And my friend Ken Griswold has, argued with me for years on some of this. And his point is, if you're looking, say, at dairy cattle, by the time you look at a herd of 1,000 head, you may only have 150 of those cattle that are going to respond to whatever your product is at any point. And I never really thought about that. So there's some animals that are just not at the right stage of lactation, that aren't genetically the right mix for this product. So you may see this improvement in the bulk tank level at a very slight difference that doesn't make that much impact on your profitability. But for those animals that it works in, it's a huge impact. So what what metric do you want to use, and how do you find the animals that it works on? And that's really part of the challenge.
Yeah, definitely. And in regards to animal species with which you've worked with, have you seen greater efficacy of certain products, or let's say in ruminants as compared to monogastrics or that kind of thing? Or do you think these products have a potential to be used in various species and be effective both from a performance standpoint, but also from assuring the quality in those foods derived from those animals?
Well, there are products that'll work in just about every species. You just have to kind of pay attention to what you're doing. You've got to acid protect some products to go through the monogastric stomach. Others you have to protect to make it past the rumen to get to the hindgut and the ruminant. Because, like, if you've got a prebiotic and you throw it in the rumen, it's not going to survive the rumen because there's so much digestigrative ability there in the rumen that most things aren't going to survive passage to the rumen. But if you can rumen protect it, then you can make changes in the hindgut, which will impact your animal performance. So as long as you're aware of what you're looking at, there's not really a species limitation. You've got to kind of target what you're doing. There's not going to be here is my lactobacillus culture that's going to work in all species all the time. That's not going to happen. We're going to have tailored lactobacillus, one strain for chickens, maybe another strain for pigs, another for finishing pigs, something different for cattle. So we're not going to be able to just say, we're not going to have the silver bullet of here's the solution, we're done. Unfortunately.
Yeah. Yeah, it's not a one size fits all approach, but it's understandable. There's physiological variations that we must take into account. So that sounds right. And so finally, Todd, could you leave us your final thoughts on the level of acceptability that you're seeing right now with these products, maybe in North America where you're working, and where do you see things evolving in the next 5 to 10 years, considering the current regulations and bans on antibiotic growth promoters and the need for the adoption of new and alternative treatments? What's your take on this, or what are some of your final thoughts regarding this?
Well, I think, like I said earlier, we're kind of in a golden age for the development of these products. And I think we're going to see in the next 5 years a lot more of these products in the market and a lot more being implemented. I think eventually it'll play out to almost be farm specific or region specific treatments for, say, chickens in the Southeast versus the ones in Arkansas have different probiotics that are used. Cattle in the feedlots versus dairy cows versus cow-calf having different products used in them. So the acceptability to most producers is, most of them will use these products. If we can tell them and show them how it's going to work, they're very progressive in being willing to try something new, as long as we can show them how and why it's supposed to work for them. So I think we'll see a lot more of these products, and especially here in North America now that they're all antibiotics are going to have a prescription to be utilized. So that's, that is going to be a lot more difficult for farmers to use products that are going to trash the ability of the probiotic, excuse me. Because we see a lot of times people will be using a probiotic and then they come back in and feed some antibiotics, and then they throw up their hands and go, well, the probiotic didn't work. Well, you dosed an antibiotic in there and you wiped out the probiotic organisms. Yeah. So of course they didn't work, because an antibiotic is going to be stronger. It's going to have more impact. So issues like that, I think, are going to make these products a lot more appealing to people. And, again, the development in these as we're coming along and understanding the microbiome now, that's the big word of the day. But as we start to understand actually what is a good microbiome for production, once we can see what that looks like and whatever our production metrics are, what we can correlate, then we can see which probiotics get close to those goals, and then we can fine tune. So I think we're going to see a lot more of that back and forth between that fine tuning as we finally develop the knowledge of what is a good or a better microbiome for production goals.
Okay. And do you see this happening through the use of like single products or more a combination of them?
Honestly, I think there's going to be a lot of multiple products being used, especially at different phases. Like I said, there's not going to be a silver bullet. So I think there's going to be a lot of room for different approaches. And I think combinationally using some of these products, like using a prebiotic with a postbiotic and a eubiotic, that there's room for using all of those at the same time in some of these species to achieve the goals that we've got.
Okay. Sounds good. Okay, Todd, I think that we'll leave it at that for now, but I want to thank you. It's been quite a nice and interesting chat. We hope to have you back for new webinars and to let us know about new developments in your research. So once again, we want to thank you for your time and for sharing your knowledge with us.
Sure. Thank you very much, Alvaro.